time to start. I only have 8 people here, but we'll go ahead and hope more people join as we go. There's just one thing I want to do today, and that is go over the questions that you submitted. Thank you very much. But these, well, let me get them up on the screen. You should be able to see areas of greatest interest.
Yes.
Yes, you can? Okay. The questions were covering a wide array of topics, many of them specific questions about certain topics. So what I've done is compile them and condense, and I've tried to address what I think are the interest, the intent of the various different questions. So I've listed here the main areas: energy, enzymes, cell division, Mendelian inheritance, photosynthesis, and oxidative reduction. So if in any of these sections, I'll go through them one at a time, you don't think that your question or questions were answered, speak up right then, because it's important that you that your questions are answered. I'm aware, and I think I told you, the average for test 3 in this class was 63%. And these were the topics that were covered in test 3. So that means that there are a lot of things that are fuzzy or that you don't understand at all, and we need to rectify that. So that's really the whole purpose of this exercise. So the first topic is energy. A number of people said that they'd like to see a review of what energy is all about. Well, the definition of energy is the capacity to do work. Well, what is work? It's anything that requires energy, including life. So if you remember from the second law of thermodynamics, life needs a constant input of energy, and if at any point that input stops, it's no longer alive. So that's why energy is so critical to a class in biology, because life depends on a constant input of energy. There are 2 types, potential and kinetic. Potential energy is stored. It's not doing anything. So for example, food, before you eat it, is potential energy. Fossil fuels, water behind a dam, they're all sources of energy, but they're not doing anything. Kinetic energy is what's working. So you eat food and then walk. That's kinetic energy. Driving after filling your car with gas, that's kinetic. Water flowing over a dam is kinetic. Cooking on your stove. So the various forms that we commonly encounter on a daily basis: heat, light, motion, electrical energy, chemical, gravitational. And these things are, for example, on the right here, some illustrations. Chemical energy in the food we eat results in motion. If you ride a bike. Radiant energy from the sun is converted by plants into chemical energy, and so on. Now, 2 points I want to make here at the bottom of this slide. Energy is either renewable or non-renewable. These two types of energy are critically important at this point in time on this planet. And we'll talk about that later in the semester if we have time. We deal with global warming and climate change. Enzymes. There were a lot of questions about enzymes, and I'll try to address them all. And if your question about enzymes isn't addressed by my remarks here, speak up right away. And one of the issues was shape. Why are enzymes a specific shape, and why are their substrates the same shape? And how does this lead to specificity, and why is specificity important? I have a diagram here which deals with the issue of shape. So on the left, you see an enzyme in gray, substrate in green. The active site has a particular shape, which is similar to that of the substrate, but not identical. However, when the substrate fits into the active site, the enzyme's shape changes so that it now fits the shape of the substrate. And when that happens, when the enzyme shifts its shape slightly, and you can see it's just a slight change, the substrate is converted to products. In other words, it changes from one green to two, one blue, one yellow. These are the products that are then released once the reaction is over. This is called the induced fit model, and it's based on the enzyme being able to change its shape after the substrate binds to it. So what's important here? Within this active site, there are various types of bond that can form between the substrate and the enzyme. And I'd like to make a distinction here between types of bond. So you recall that an enzyme, in order to be active, has a tertiary form. And that form is held in place by bonds, hydrogen bonds, ionic bonds, hydrophobic interactions. So the enzyme is configured by bonds. Now, in addition to that, when catalysis is underway, other bonds form between the enzyme and the substrate. And it's the formation of these bonds that is critical for catalysis. So I'll read this sentence again. Within the active site, various types of bond can form between the substrate and the enzyme. Weak bonds form first. So that would be, for example, hydrogen bonds and hydrophobic interactions. So when the substrate first enters the active site, you get the formation of these weak bonds, followed by formation of stronger bonds, for example, ionic and covalent. So the differential force that occurs between the strong and the weak bonding causes torsion, which is twisting. This is what breaks or weakens the bonds within the substrate. Oh, some of you asked, what is the force that actually causes catalysis? And it is, at least in a major way, this differential force between the strong bonds and the weak bonds that form between the substrate and the enzyme active site. So
I've got a question, Professor, on
yeah.
this.
Uh-huh.
On the weak bonds, you can see where you're saying where the differential can cause it to break and weaken.
Uh-huh.
On the stronger ones, the ionic bonds, the covalent bonds, can those break as well, or only the weak ones?
We're talking about bonds within the substrate. Okay, so the substrate has bonds that are holding it together. This green object here is held together by chemical bonds. And some of those have to break in order for the substrate to be converted to products. So the question...
You answered my question already.
Okay, all right. It is a good question. So it raises the issue. There are really three types of bonding that we're talking about here. One is the bonding that holds the substrate together. Another is the bonding that holds the enzyme together. And the third is the bonding between the substrate and enzyme that forms upon the entry of the substrate into the active site. And if we go down to this orange diagram, the point here is to illustrate that type of bond, this third type of bonding between the substrate here in blue and the enzyme active site here in orange. And what's shown here are a hydrogen bond and two ionic bonds. Could also have hydrophobic bonds or even covalent bonds. And it's the formation of these bonds that causes catalysis to go forward. The caption here says the substrate is held in the active site by a variety of bonds, hydrogen, electrostatic interactions, etc.
Is there like a minimum? Could it be like just two or?
I don't know if there's a minimum. Well, there have to be at least one.
Yeah, it's set.
But I don't know what would be the maximum.
Yeah.
It would probably depend on the
Substrate.
the particular interaction.
Yeah.
Some would probably have more than others. But what's important is that bonds do form and that they have different strengths, and that differential in strength of bonding is what causes bonds in the substrate to break or weaken. Okay, are we okay so far?
Yes.
Okay, now the importance of specificity. Well, within a cell, there are thousands of enzymes, and each one has a specific substrate. Enzymes, in any given enzyme, doesn't have more than one substrate unless it just happens to be converting two substrates to one. And so why is that? Why do we have thousands of enzymes in order to control thousands of reactions that are going on in our cells all the time? Well, when you think about it, if it wasn't for specificity, so supposing you had one enzyme could catalyze two different pathways. Now, if your body needed one of those pathways and not the other, since the same enzyme is catalyzing them both, they'd both be running. If one was running, they're both running. So you can't have differential control of metabolism unless you have this extreme specificity between enzymes and their substrates. So does that make sense? Okay. I guess it makes sense. There were a number of questions about enzyme denaturation. Now this picture you have seen before. Enzymes can be damaged permanently or temporarily by exposing them to extremes of temperature, pH, or salt concentration. This is denaturation. And what happens is the tertiary structure is destroyed so that the enzyme can't function properly. Well, that's illustrated in this little diagram. On the left, a normal protein, tertiary structure, folded with loops and helices and so on. If its environment is extreme, caused by, say, a change in temperature, this falls apart, and you get, on the extreme condition, you get a denatured protein that has no tertiary structure at all. It's just this long chain of amino acids. So that has no enzyme activity. It's denatured. In some cases, a denatured protein, if you restore the environment, it can renature back to a normal protein and have activity again. But that's relatively rare. Among most enzymes, once they're denatured, they're done. I've tried to illustrate that in a more real way in this illustration, where the green line is a protein, an enzyme, with its tertiary structure intact. It's functional. And you'll notice these three little colored balls. Those are the active site. They illustrate the active site. Now, the substrate is shaped in such a way that it fits over these three little balls. That would be, it fits into the active site. Now, if this protein is denatured so that it loses its tertiary structure, look what happens to the active site. Now it's distributed in a completely different array, and the substrate has no place to bind. And again, on the side here, I remind you that the bonds for tertiary structure include, so that's the structure for the one on the left, include hydrogen bonds, hydrophobic interactions, ionic bonds, disulfide bridges, and there are some other weak ones, but there are a variety of different bonds that hold the protein into a tertiary structure, and denaturation causes these bonds to break. Of course, the weakest ones would break first, but it doesn't really matter. I think the breakage of any bonds in a tertiary structure would denature the enzyme. Some of you talked about difficulty understanding graphs. So here is one with the rate of reaction on the y-axis, temperature increasing from 10 degrees to 60 degrees centigrade on the x-axis. And so, and we're measuring the speed with which substrate is converted to product. And that rate increases with temperature up to a point, up to about 47 degrees. And at that point, it stops increasing, and any warmer than that, it starts to go down. And the reason it starts to go down is that at 47 degrees, the enzyme starts to denature. The temperature is too hot, the bonds start to break, and it starts to fall apart. If that were not the case, if we're not denatured, then this line, the upward trend of this line, would continue upward, because as temperature increases, the reaction rate increases. It would increase further if it could, but it can't because it's too much for the enzyme. It denatures.
So Professor, I think there was a question like this before where it was the A, B, and C, and there was the confusion on what B and C was. The B and C was on the right-hand side of the line. I think it was on one of the tests. So if it's both are on this right-hand side of the line, you're going to pick the one that's higher because it's closest to the top of the hill, so to speak. If there's 2 points on that end of that.
Oh, one on a red arrow and a blue arrow. Is that the one you're thinking of?
There was that one too, but I know there was one that had like the A, B, and C points.
Oh, 1, 2, and 3. Yeah.
But the red arrow and the red and the blue arrow were similar. So if there's 2 on the red side, we would always pick the one on that's highest because it's closest to the top.
For which question? You know, are we asking where is denaturation occurring?
Yes.
Yeah. Well, in this case, it would be on the right. Denature in the red area of this area under the curve. The red area would be the area of denaturation.
Professor, I think I remember the 3 questions, if that'll help.
Yeah.
The first one was, what is needed to start the effect, and that was energy, I believe. The second was, what was the top point of the hill? And then the second was, third one was, what starts to happen at the 50?
I think it's question number 2.
Yeah, I don't have the quiz in front of me right now.
Yeah.
But I think I'm recalling that the number 1 arrow was pointing. I said, what was happening at number 1? It had to do with enzyme activity is increasing. I think that was number 1.
At the
Number 2 was that's the optimum temperature.
Yeah.
And number 3 was the denaturation.
Yeah.
Yeah, I vaguely recall it now.
Thank you.
Okay, any other questions about this graph or the reading of graphs? There were several questions about activation energy. In particular, what is it? Well, the definition is it's the minimum energy required for a reaction to occur. Catalysis is the increase in the reaction rate that occurs by lowering the activation energy. So there are a couple of key points here. I bolded one of them. The source of activation energy is typically heat from the surroundings. In other words, if you have a reaction mixture in which a reaction is going on, the temperature of that solution, that's where the heat comes from for the activation energy. So this graph illustrates activation energy in the presence and absence of a catalyst, in this case an enzyme. So the long double-headed arrow, the red arrow, shows how much activation energy is needed without an enzyme. think heat. So this much heat is needed to convert substrate to product without an enzyme. The green line shows with an enzyme, and you can see that the amount of heat required to make the reaction go is much less with the enzyme. So we have two key points to be made here at the bottom. The source of activation energy is heat, and the energy for enzyme catalysis is torsion, the twisting that results from this differential strength of bonds between the enzyme and the substrate. So I hope this helps to clarify the mechanism. What is the source of energy? There are the sources of energy. There are two for a reaction to occur. One is the activation energy, which is heat, and the other is within the enzyme itself, the differential strength of bonding between the enzyme and the substrate. So that's the green line, and the purple line is the additional amount of heat that's needed if you don't have an enzyme. So remember these two key points about what makes a reaction go. Any more questions about activation energy or how catalysis works? There's several questions about types of enzyme inhibition. So I'll show you a couple of illustrations that you have seen before. First, we'll talk about reversible enzyme inhibitors that can be either competitive or non-competitive. What do those terms mean? Make this a little bigger. A competitive inhibitor mimics a substrate. So you have this blue object, which is the inhibitor. The yellow is the substrate. Their shapes are roughly the same, but not identical. The enzyme is illustrated by this blue object, and either the substrate or the inhibitor can fit into the active site. Active site is the right shape for either. If the inhibitor fits in it, occupies that site, there's no room for the substrate and there's no reaction. You can think of this as, for example, supposing there's a drug to treat a disease. And supposing the disease is caused by this enzyme converting this yellow substrate to a product. Now, if some drug company can manufacture a molecule that is similar in shape to the natural substrate that will fit into the active site, and then if you have a lot of this drug in the system, all the active sites will be occupied by the drug and no substrate is converted to product, and it inhibits the disease. The same thing can happen, but non-competitively. That's shown on the lower right. Again, we have the yellow substrate, we have the active site. The substrate can fit into the active site, but there's another substance called a non-competitive inhibitor that has a binding site in the enzyme, not in the active site, but somewhere else. And if it can fit into that site, it doesn't compete with the substrate, but it does change the shape of the enzyme so that now the active site no longer fits the substrate. And again, the reaction doesn't occur because this inhibitor is there. And again, you could think of, if you want to think of real life example, think of a drug for treatment of some disease. And the disease is caused by this reaction. The reaction between the enzyme and the substrate causes the disease. The inhibitor stops it. Okay. Then there's another completely different type of inhibition. And again, these are, all of these types of inhibition are how metabolism is regulated. Could you mute your mic, please? Thank you. All of these different types of inhibition are ways that cells use to control their metabolism so that all enzyme reactions aren't running at the same time. And feedback inhibition is another one. Feedback inhibition occurs when there is a pathway with several steps, each step catalyzed by a different enzyme that ends up with a product. And this is called the end product. And that end product has some use in metabolism. So as long as the metabolism, the homeostasis of the cell, needs that product, it will be swept away and used as fast as it's made. But if there's no longer a need for the product, it has no place to go and it will accumulate. And that accumulation could be lethal if it's not stopped. So in the case of feedback inhibition, this enzyme number 1, the first one in the pathway, has a site to which this end product can bind. And if the end product binds to that site, the enzyme is inactive and step 1 does not occur. If step 1 doesn't occur, none of them do. So we have the entire pathway is shut down if the first step is shut down and no end product accumulates anymore. Now, if someone still has their mic on, could you turn it off? Can't tell who it is. But hopefully you heard me and have muted your mic. So this is a dynamic process whereby the pathway is constantly monitoring the quantity of this end product. And if the quantity is low, that means it's needed somewhere and the pathway runs. If the quantity of the end product is high, it means it's not needed, and it can itself shut down its own biosynthesis. So is that clear about feedback inhibition? Well, those are reversible types of inhibitions. Inhibition can also be irreversible.
That's not my nature.
We still have somebody's mic on. Kiyomi, is it yours? Now it's off. Okay, thank you. Irreversible inhibition occurs if the inhibitor is covalently bound to the enzyme. So you see that here on the left. The enzyme is in purple, the inhibitor is in green, and they're bound together covalently, so they can't come apart. And if the substrate comes along, even though it's got the right shape, to fit into the active site. It can't because the inhibitor is there. And the inhibitor will never go away because of this tight bonding. An example of that is the way penicillin works. Penicillin is a small molecule. The structure of it is shown here in these 3 figures. It's it's called a beta-lactam. And what it does, it's antibacterial. And the way it works is it binds to this enzyme, transpeptidase, that's required for the bacteria to make cell wall. So if penicillin is not there, the transpeptidase is involved in wall synthesis and the bacterium is healthy. If penicillium is there, it binds covalently to the enzyme and never comes off, and the bacterium has no chance to make a cell wall. So that's shown here. I've circled the covalent bond between penicillin and the enzyme. The enzyme is shown in purple. Okay, so that's an example of irreversible inhibition. And you want, for example, in the pharmaceutical industry, there would be many times that you would want irreversible inhibition so that your treatment for a disease is permanent. Some people asked about zymogens. They are inactive forms of enzymes. And this picture illustrates a zymogen and what happens to it. So the enzyme is illustrated here in this blue object. It is in the form of a zymogen because the red part covers up the active site. So the substrate can't fit in there because the site's covered up by a component, which if it's removed, now the enzyme is active and the substrate can fit into the active site. So I hope that is a picture in your mind of how a zymogen works. And a couple of people asked, are zymogens? We talked about zymogens in the pancreas and how zymogens are made. Zymogens of proteases are made in the pancreas because if they were active, they'd digest the pancreas. So they're made as a zymogen, and when they get out of the pancreas and into the intestine, there are enzymes that remove the part that's covering up the active site, and in the intestine, the enzyme is active. Now, are there any other places in the body where there are zymogens? I could only find an example of one, and that is salivary glands. Also, in plants, carnivorous plants, that would be, for example, plants that eat insects, they apparently have proteases that are produced as zymogens. But they're not very common, and the only place that we have a good example of them is in the pancreas. And to emphasize a point, zymogens are not denatured enzymes. In other words, if you go back to this figure, this zymogen has a full tertiary structure. It's ready to go. It's not denatured. The only problem it has is this part that's covering up the active site that can be readily removed by another enzyme. So this is not denatured. It's an active protein. A denatured enzyme would be where this thing has fallen apart, has no tertiary structure left. Any questions you have about enzymes, because I'm going to leave the topic of enzymes now and talk about cell division. Several of you ask about mitosis versus meiosis, similarities and differences. For that, I refer you to this worksheet, and I've copied a picture out of the worksheet, which has processes that are unique to mitosis, some unique to meiosis, and some which happen in both. So which one has two cells result from it? It's mitosis. You see that here, mitotic cell division. One cell becomes two cells. Whereas in meiosis, a division results in four cells. And you can see that this is a fuzzy illustration, unfortunately, but you start with one cell, and if you go around to meiosis 1, followed by meiosis 2, you end up with 4 cells. So it's a 1 division process in mitosis, it's 2 divisions in meiosis. Mitosis produces somatic cells, whereas meiosis produces gametes or reproductive cells. In the case of mitosis, the 2 cells that are produced here are identical to each other and identical to their progenitor, to the starting cell. So this cell is reproduced exactly as 2 cells. In meiosis, quite the opposite happens. You start with the first meiotic division, you get 2 haploid cells. These haploid cells undergo the second meiotic division, and you end up with 4 cells, each of which is unique. So none of them is like any of the others, and none of them is like the parental cell, the cell that started. And that's because variation has been introduced. We can talk about that in a little bit. Mitosis is asexual, whereas meiosis is sexual. And in meiosis, homologous chromosomes pair. And I'll deal with that in just a bit. Now, these are all differences. What are the similarities? Well, in both cases, DNA replication has to happen. You can't have a cell division if you don't duplicate the DNA, replicate the DNA. Both of them occur in eukaryotic cells. In both cases, sister chromatids are produced, and in anaphase, both mitotic and meiotic anaphase, the chromatids separate. And in both cases, during division, the chromatin condenses so that you can see it in a microscope, visible structures. And both have common phases in the process of dividing: metaphase, telophase, prophase, and anaphase and cytokinesis. Okay, any questions? If you need to refresh your memory about specificities or commonalities, just refer to this. One of the points about meiosis is that it produces gametes. And we'll talk about gametes later when we deal with inheritance. But I refer you to this figure, which illustrates gametes. So this is a human life cycle. Humans spend most of their existence as diploids, but within the ovary in females, within the testis of males, meiosis occurs. And if it's a female, the gamete that is produced is an egg. If it's a male, the gamete that is produced is a sperm. So these are necessary. These are haploid cells, these gametes, and it's necessary for them to get together in order to start the life cycle over again. And that's called fertilization, when a sperm enters an egg. So now you have the diploid phase, and it's called a zygote. And following this, every cell division is mitotic. So for the rest of an individual's life, all the cell divisions are mitotic except for those that go on in the ovary and the testis. Now we talked about crossing over and recombination. That occurs only in meiosis and only in meiosis 1, in the first mitotic division, where the chromosomes from the homologous chromosomes come together in a very tight association, so tight that they can exchange parts, and they do. They change a lot of parts in real life. What this shows is just one exchange, so you start out with a blue chromosome and a red one. They make this so-called tetrad, and when they come out of that, there's one chromosome, the red one, now has a red half. The other half is red with blue tips. The corresponding blue chromosome has one original and one recombinant, is blue with red tips. So this is a source of variation that occurs only in meiosis 1. And I included this slide to contrast that with independent assortment, which occurs twice in both meiosis 1 and meiosis 2. In both cases at metaphase, metaphase 1 and metaphase 2, the chromosomes line up and the direction that they go is entirely random. So you can either end up with the same configuration as they started with or different. Any questions about that? A couple of people asked about the role of cell division in evolution, whether it's mitotic or meiotic. Well, mitosis is needed just to keep the organism alive and developing properly. So mitosis is needed for growth, to repair wounds, and for exact reproduction. You need, for every cell in an adult body, which is no longer developing, you have to have exact reproduction in the diploid. A meiosis is needed to generate variation. Natural selection requires a highly varied pool of germplasm in order to select the fittest organisms at that point in time for that environment. But the other goal of meiosis in evolution is to generate gametes, shown here. Female gametes and male gametes, which are necessary in order for sexual reproduction to occur. Some people asked about cytokinesis, what's different in plants and animals. So these are pictures that you have seen earlier. In animals, there's no cell wall. And the cell needs to be divided into two, and that's done by this contractile ring, which pinches the cell in the middle and simply squeezes it into two subsequent cells. That can't happen in plants because of the cell wall. So what happens in plants is that the components of the cell wall are assembled in vesicles in the cell that's already undergone telophase. And these vesicles will start to fuse and make what's called a cell plate. And this plate then continues to produce vesicles, which will allow it to fuse to the walls of the new cell. And when that process is complete, there are now two daughter cells. All right, now I want to deal with that extra credit worksheet that you had. Most of you got most of everything right, except for the last page, page 5. And the reason you had trouble with page 5 is that you were really unprepared for it. You had to study the diagram on page 5 very carefully to try to understand how to answer the questions. But let me tell you the point that page 5 was making. It was spermatogenesis versus oogenesis. So production of sperm versus production of eggs. Oh, let me make this a little bigger. In the case of sperm, you start with a spermatogonium, the original cell, and you have meiosis 1, which gives you 2 haploid cells, and then meiosis 2, which gives you 4, ends up with 4 haploid cells. And each of those haploid cells is a viable sperm. So spermatogenesis gives you the 4 products of meiosis are all alive and well. In the case of egg production, you start with an oogonium. You have meiosis 1, but now what's called a secondary oocyte is a viable cell, but the other product of that meiotic division is not viable. It's called a polar body, and it dies. Does not divide again. This one that's still alive does divide again in meiosis 2, and it makes 2 products, but again, only one of them lives, the ootid. The other is a second polar body, and it dies. So that you end up in females making each meiotic division, or each set of meiotic divisions, ends up with just 1 gamete, just 1 egg. Males end up with 4 gametes, females end up with 1, and that's because 2 of them are lost as polar bodies. So is that clear, the distinction between sperm production and egg production? And you had to use that information to answer the questions on page 5. So there's only one question about sperm production. Meiosis produces 4 functional sperm cells in spermatogenesis. So that's shown in the first diagram at the top of that page. Then oogenesis is different. Meiosis begins in the embryo and is arrested in prophase 1. Cytokinesis is unequal. In other words, when the cytoplasm divides, it's unequal. You end up with 1 viable oocyte and the other is a polar body. which dies. In meiosis II, it's completed when the secondary oocyte is fertilized, and cytokinesis is, again, unequal. It produces the egg, it's the final product. The other product is a second polar body, which dies. So at birth, the ovary of a female contains a lifetime supply of primary oocytes, that's eggs, that are arrested in prophase 1 of meiosis. So the point I want to make here is that you understand the distinction between making sperm and making eggs.
Sorry, I've got a question on that.
Uh-huh.
Is there a particular reason why the secondary polar bodies and the female die compared to the males?
It's a good question, and I don't know the answer. Why, evolutionarily, why that has resulted in the death of so many egg cells, potential egg cells. Whatever the reason, it seems to be working. Okay, now I want to get through Mendelian inheritance. I'm not going to be able to finish this today. We'll finish this set of questions and answers on Wednesday, but there were quite a few questions about inheritance and how you identify gametes. Several questions were about how do you do a dihybrid cross? I emphasize you should know how to do this. Many of you don't know how to do it, or didn't, at least in test 3. It's important that you learn, and the best way to learn how to do this is sit down with a pencil and paper and draw these things out for yourself. And you have plenty of examples of problems on a worksheet that was not graded. I just sent it to you as a list of problems. So let's take this dihybrid cross, which I think, I can't remember, but I think you've seen this before. And you have to read the issue carefully. A short stem, so there are two traits here, a short stem and a yellow pod plant is crossed with a true breeding long stem green pod. So the two traits are stem length and pod color. You are told that long stems are dominant to short stems. So long is dominant, and green pods are dominant to yellow ones. And the question asks, what are the genotypes and phenotypes of the F1 and the F2, which is stated here as a cross between two F1s. So that would be an F2. Well, the first thing you have to do in a question like this is assign symbols for the traits. The traits that you see are the phenotypes. You need symbols for the genotypes. So I, for this question, arbitrarily chose L for stem length. And we're told that long stems are dominant. And so we assign a capital L for long. Now that means since the other stem length is recessive, that it would be lowercase l. Similarly, I chose the letter G for green pods, and it's a capital G because we're told that green pods are dominant. Therefore, the yellow pods are little g. And some of you have, for example, in the case of pod length, you'd say the dominant allele is G, the recessive allele is Y. That is not right. That will get you in trouble every time. In fact, it makes it impossible to solve. Okay, once you've assigned gene symbols, letters, you look at the phrase true breeding. Now, what does true breeding mean? Many problems like this have that word combination, true breeding. So you need to know what that means.
Homologous.
Yeah, it means that all loci, or at least the loci that you're concerned with, are homologous. And what does homologous mean?
The same genotype.
The same, that both alleles are alike. These plants now have been inbred so that at every locus, or at least the two loci that we're looking at, both alleles are alike. So long stem is dominant, and we can call it, it's true breeding, so that it's homologous. Big L, which makes short stem recessive, little l. And similarly with pod color, green is dominant, and it's true breeding, so it's homologous, so it's big G, and yellow is little g. And so the parents now, we know, are both homozygous, homozygous, big L for the dominance, and little g for the recessive parent. Now, what gametes? Several of you ask, how do you determine gametes? Well, there's this method called FOIL, first outside, inside, last. If you use that, you can see first are big L, big L and little l. Outsides are big L, little g. Insides are big G, little l. And lasts are a big G, little g. So the gametes that can be produced by the parents are big L, big G, and little l, little g. So the F1 then would be all heterozygotes. You cross two homozygotes, the F1 is all heterozygotes. And so the phenotype then, the answer to that question, the phenotype is long stem and green pod. Now, what gametes does this F1 produce? Well, going through the same process, the gametes are either homozygous dominant, big L, big G, or homozygous recessive, little l, little g, or heterozygous, big L, little g, little l, big G. And now we're crossing an F1 with an F1. So they both have the same set of gametes. So you do your Punnett square by plotting one set of gametes on one axis and identical set of gametes on the other axis. And then you cross, you do the combinations. So this first square is big L, big G, big G. You can go on down and make all the right combinations. And then you ask, so those are the genotypes. And then you can ask, what are the phenotypes in each one of these boxes? Well, if you list out all the genotypes, you can see that there are different numbers of the different genotypes. And all of these numbers, you add them up, it comes to 16 because you have 16 boxes. And when you report the phenotypes, you simply count what's in each box, what it looks like. So there are 9 boxes that are long and green. 3 boxes are long and yellow. 3 are short and green, and there's only 1 that's homozygous recessive, short and yellow. So that is the way you deal with a dihybrid cross. Any questions about how you work your way through this? Reading the question carefully, and then choosing gene symbols, making sure that you indicate dominance and recessiveness correctly, and then name the gametes. Gametes from the from the parents and gametes from the F1. All right, there were some questions about identification of gametes, and this problem illustrates well how you do that. The question here was, a homozygous parent is crossed with a heterozygous parent, and it gives you the genotypes of the homozygous parent, big F, little g, that's a homozygote. And the heterozygous parent is big F, big G, little g. So what gametes are produced by the homozygous parent? The homozygous parent only has big Fs. There's no little f's. It has only little g's, no big G's. So the only gametes that it can produce are big F and little g. So that's the answer to the first part of the question. Gametes from the homozygous parent are big F, little g. And what are the gametes produced by the heterozygous parent? Well, now there are more choices. Again, you can use that FOIL method or just look at them. But one possible combination is big F, big G here, or big F, little g here, or little f, big G, or little f, little g. So those are the 4 possible gametes. So you see how we arrive at that. That's the answer to the second question. Those are the gametes. Then the third part of this is, how many boxes would you need in your Punnett square to account for all the possible gamete combinations? Well, the key here is to recognize that the homozygous parent can only produce one type of gamete, big F, little g. So if you're making a Punnett square, you say on one axis, we'll put that gamete. The other parent made 4 different types of gamete. So you list those on the other axis, and then you do the matching, and you see that what you get are these 4 products. So the answer to how many boxes do you need, the answer is 4. Why don't you need 16? Because it's redundant. Since you have only one type of gamete from this parent, you can plot that gamete 4 times across this axis, but it's the same thing every time. You get exactly the same thing every time. So that answer, and some of you said 16, that's not wrong, but it's not the simplest way to do this. And what it reflects is that you weren't really thinking carefully about what the question is asking. And I have up here in the upper right the type of Punnett square that I was expecting, where you'd have just 4 boxes. So is that clear? The main point here is how to choose gametes. How do you know what your gametes are? Once you know the gametes, you can determine the outcome of a Punnett square. Okay, I've run out of time. So the rest of your questions, we're down, we finished Mendelian inheritance. We still have photosynthesis to do, oxidation, reduction, reactions, chemiosmosis. Those things we will do next time on Wednesday. And once we finish that, then we'll look at a slide set on respiration. So, yeah.
Can I talk to you after class?
Yeah, I'll leave it on. Anything else? Did you find this helpful at all?
Very.
Very helpful, yeah.
Yes, very helpful.
Informative.
Okay, good.
Yeah, I really appreciate it.
All right, well, I'll have to keep that in mind, maybe for in the future for a particular combination of topics. We can do the same thing. I appreciate the questions that you ask because it gives me insight into how you're perceiving some of the things I'm presenting. All right, well, time's up. Mostly I'll leave the Zoom on, and that's it for everybody else.
Thank you, Professor.
Okay, see you Wednesday.
Just a quick question on the extra credit is, will that get the score get adjusted in Canvas?
Yeah.
After it's okay.
Yeah, so there are 2 things there. One is that that extra credit exercise, it was presented to you as an assignment. So you get a score for whatever number of answers you got correct. Then at the end of the semester, the extra credit will be your score added on to your total for the semester.
Okay. Got it. Thank you.
Uh-huh. Okay, anything else?
I've just got a question about the group project, but I'll go after the other person who had a question.
Okay. All right, we'll wait. Patrick and Boschley.
Okay, so I have a questions on the extra credit. Do you see my email? I emailed my worksheet to you on Thursday, I think.
I think so. The
Yeah, because every time I submit it on Canvas, I don't know why, but you can't see my answer or anything at all. The
yeah.
document it's not coming up.
Okay. Can you submit it to me by email?
Yes, I did on last week.
All right. Well, let me check. I may have it. And have you looked to see if you've received a grade for it?
No, I don't.
This is for the extra credit, right?
Yes.
And you don't have a grade.
Let me check to making sure because I checked last time and I don't have grade. No, I don't have grade.
Okay, I will see if I can find, you sent it to me by Canvas email or personal?
I need to check because I sent it by your campus email.
Campus email, okay. Well, I will look that, and if I don't have it, I will let you know.
Okay, thank you.
Mhm.
And I got another question. So last week you attached the email from my group member. I think his name is Andreas. I don't know his name. So I sent the email to him back a couple of days, like on Saturday, I think, and I have not get any response from him yet.
Okay, so you have one of your members has responded, but the other one hasn't? Is that right?
Yeah, he said he's still doing. Yeah, he said he's still doing the group project, but I emailed him to ask him about, oh, what should we do? We need to meet on the Zoom or something like that? But I have not get any response from him yet.
Okay, I will send a message to all of you indicating that there has to be a response by Wednesday this week, or we go on without that person. So you should get that from me.
So if he's not getting so
Yeah,
if he's not respond to any of my email or member email, can I go without him, or I have to wait for him
no,
to actually work together?
no, if we don't hear from him by Wednesday, then we go on without him. There are other groups that have missing members. And so what we may do is reassign group membership, but at the very least, you go on without the missing member in your group. And
Okay,
we'll make that
thank you.
decision on Wednesday. Either we hear back by Wednesday or we go without.
Without him. All right. Thank you. Have a good one.
Okay, take care. Okay, Patrick, what's up?
Yeah, so I just was a little confused on my group's mutant. Our mutant is R553X. And when I'm initially looking at it on the CFTR2 website where you look it up on there, it says this variant causes CF when combined with another CF causing variant. So I guess my question is, do I need part of my project? Should I include what that other variant is or would be? And do I kind of need to include that in the project, or am I just only doing the mutant that I have?
Well, are you saying that your mutation alone causes no change, does not cause cystic fibrosis?
That's what it looks like, because it says this variant causes pancreatic insufficiency when combined with another variant that causes pancreatic insufficiency.
Okay, so that is dealing with one symptom. It doesn't say anything about lungs. So you'll have to look further in the literature, and don't depend entirely on the websites that are given there. Look on the web, and you might have to resort to primary literature. And the best way to do that is to go into the National Library of Medicine.
Okay. Yeah, that's where I'm at, actually. I've got a few peer-reviewed things open.
Yeah, you're on the right track then.
Okay. Okay. All right. I think that's it.
Okay. Well, that would be, I'd be surprised if that mutation alone does not cause something.
Right.
Otherwise, it wouldn't be on the list.
Okay.
All right.
Thank you.
Yeah. Okay. Take care.
You too.
Okay. Bye.
Bye.